In the previous module, we discussed nitrogen cycle. Nitrogen fixation is the first step of the nitrogen cycle. It is a process in which nitrogen in the atmosphere is converted into ammonium or nitrogen dioxide. The growth of all organisms depends on the availability of nutrients, and none is more important than nitrogen, which is required in large amounts as an essential component of proteins, nucleic acids and other cellular constituents. There is an abundant supply of nitrogen in the Earth's atmosphere, nearly 79% in the form of nitrogen gas. However, nitrogen is unavailable for use by most organisms because atmospheric nitrogen or molecular nitrogen N2 is relatively inert and does not easily react with other chemicals to form new compounds. The stable nature of nitrogen is because of the triple bond between two nitrogen atoms, making the molecule almost inert. In order for nitrogen to be used for growth, it must be fixed or combined in the form of ammonium or nitrate ions. The fixation process frees up the nitrogen atoms from their triple bonded diatomic form to be used in other ways. Nitrogen fixation, natural or synthetic, is essential for all forms of life because nitrogen is required to biosynthesize basic building blocks of plants, animals and other life forms. For example, nucleotides for DNA and RNA and amino acids for proteins. Therefore, nitrogen fixation is essential for agriculture and the manufacture of fertilizers. It is also an important process in the manufacture of explosives, for example, gunpowder, dynamite, TNT, etc. Nitrogen fixation takes place mainly in two ways: biological or non-biological. Non-biological nitrogen fixation is carried out by either natural nitrogen fixation, that is by lightning and ultraviolet radiations, or industrial nitrogen fixation, that is by Haber's process. The third type is biological nitrogen fixation, that is carried out by microbes. Some estimates of the amount of nitrogen fixed on a global scale can be seen in the table. It is evident from the data that the total biological nitrogen fixation is estimated to be twice as much as the total nitrogen fixation by non-biological processes. If nitrogen fixation is done industrially, large input of energy is required and is carried out at 600 degree centigrade temperature and a pressure of 200 atmosphere. However, microbes do it at room temperature and at 1 atmospheric pressure. Industrial nitrogen fixation by Haber's process. The possibility that atmospheric nitrogen reacts with certain chemicals was first observed by Desfosses in 1828. He observed that mixtures of alkali metal oxides and carbon react at high temperatures with nitrogen. Haber's process was discovered in 1909 and is still used by fertilizer industries to fix atmospheric nitrogen into ammonia. The process converts atmospheric nitrogen to ammonia by a reaction with hydrogen using a metal catalyst under high temperature and pressures. It is a reversible process in which nitrogen and hydrogen combine to form ammonia. Hydrogen and nitrogen are mixed in a ratio of 3:1. Because the reaction is reversible, not all the nitrogen and hydrogen will convert to ammonia. The original Haber-Bosch reaction chambers used osmium as the catalyst, but it was available in extremely small quantities. Later on, a much less expensive iron based catalyst was proposed, which is still used today. The most popular catalysts are based on iron promoted with K2O, CaO, SiO2 and Al2O3. Artificial fertilizer production is now the largest source of human produced fixed nitrogen in the Earth's ecosystem. Ammonia is a required precursor to fertilizers, explosives and other products. The Haber process requires high pressures around 200 atmosphere and high temperatures at least 400 to 500 degrees centigrade and other routine conditions for industrial catalysis. Much research has been conducted on the discovery of catalyst for nitrogen fixation, often with the goal of reducing the energy required for this conversion. However, such research has thus so far failed to even approach the efficiency and ease of the Haber's process. Biological nitrogen fixation. Biological nitrogen fixation, often called BNF, include conversion of nitrogen to ammonia by microbes. All biological nitrogen fixation is done by nitrogenase enzyme complex which contain iron, molybdenum or vanadium. Microorganisms that can fix nitrogen are prokaryotes called diazotropes. In other words, diazotropes are the organisms which use N2 as their sole source of nitrogen. Some higher plants and some animals have symbiotic association with diazotropes. All the nitrogen fixing organisms are prokaryotes. Some of them live independently of other organisms, that is the so called free living nitrogen fixing bacteria. Others live in intimate symbiotic associations with plants or with other organisms, for example, protozoa. Biological nitrogen fixation was discovered by the German agronomist Hermann Hellriegel and Dutch microbiologist Martinus Beijerinck in 1901. Biological nitrogen fixation occurs when atmospheric nitrogen is converted to ammonia by an enzyme called nitrogenase. The reaction for biological nitrogen fixation is nitrogen N2 plus 8 protons plus 8 electrons, giving ultimately 2 molecules of ammonia and H2. The process is coupled to the hydrolysis of 16 equivalents of ATP and is accompanied by the co-formation of 1 molecule of hydrogen. Thus, biological nitrogen fixation can be represented by the following overall equation, in which 2 moles of ammonia are produced from 1 mole of nitrogen gas at the expense of 16 moles of ATP and a supply of 8 electrons and 8 protons. The enzymatic reduction of dinitrogen to ammonia requires both a reducing agent such as Ferredoxin or Flavodoxin and an input of chemical energy released from the hydrolysis of ATP to overcome the activation energy barrier. The conversion of nitrogen to ammonia occurs at a cluster called FeMo-Co, an abbreviation for the iron molybdenum cofactor. The mechanism proceeds by a series of protonation and reduction steps, wherein the FeMo-Co active site hydrogenates the nitrogen substrate. The reactions occur while nitrogen is bound to the nitrogenase enzyme complex. The iron protein is first reduced by electrons donated by Ferredoxin. Then the reduced iron protein binds to ATP and reduces the molybdenum iron protein, which donates electrons to nitrogen, producing NH double bond NH. In 2 further cycles of this process, each requiring electrons donated by Ferredoxin, NH double bond NH is reduced to NH2 NH2, and this in turn is reduced to 2 molecules of ammonia. Depending on the type of microorganism, the reduced Ferredoxin, which supplies electrons for this process, is generated by photosynthesis, respiration or fermentation. In free living diazotropes, the nitrogenase generated ammonium is assimilated into glutamate through the glutamine synthase or glutamine synthetase pathway. Measurement of nitrogenase activity. As with many assays for enzyme activity, it is possible to estimate nitrogenase activity by measuring the rate of conversion of the substrate nitrogen to the product ammonia. Since ammonia is involved in other reactions in the cell, it is often desirable to label the substrate with 15N to provide accounting or mass balance of the added substrate. A more common and popular assay is the acetylene reduction assay or ARA, which estimates the activity of nitrogenase by taking advantage of the ability of the enzyme to reduce acetylene gas to ethylene gas. These gases are easily quantified using gas chromatography. ARA has been applied to a wide range of test systems, including field studies where other techniques are difficult to deploy. So far, all samples containing nitrogenase were found to give positive ARA test too. Properties of nitrogenase enzyme. A unique property of this enzyme is its sensitivity to oxygen. The enzyme is inhibited by the presence of oxygen. Nitrogen fixing bacteria have different strategies to reduce oxygen levels which interfere with nitrogenase function. Enzymes responsible for nitrogenase action are very susceptible to destruction by oxygen. Due to the oxidation carried out by oxygen, most nitrogenases, which are essential large reduction complexes, are irreversibly inhibited by oxygen, which degradatively oxidizes the FES cofactors. In essence, oxygen binds to the iron found in nitrogenases and blocks its ability to bind to nitrogen. This requires mechanisms for nitrogen fixers to protect nitrogenase enzyme from oxygen in vivo. Although oxygen sensitivity of nitrogenase is not a problem for anaerobic bacteria, it could be a major problem for the aerobic species such as Cyanobacteria, which generates oxygen during photosynthesis, and also for the free living aerobic bacteria of soils such as Azotobacter and Biserinckia. These organisms have various methods to overcome the problem. Some microbes have a proteoglycan rich extracellular matrix which traps a layer of water, often referred to as a slime layer. This slime layer acts as a barrier for oxygen. Some nitrogen fixers such as Azotobacter species have the highest known rate of respiratory metabolism of any organism, so they might protect the enzyme by maintaining a very low level of oxygen in their cells. Many rhizobia, that is nitrogen fixing bacteria, live in a symbiotic relationship with plants known as legumes. They have an interesting strategy to deal with oxygen. In plants infected with rhizobium, for example, legumes such as alfalfa or soybeans, the presence of oxygen in the root nodules would reduce the activity of the oxygen sensitive nitrogenase. In these situations, the roots of such plants produce a protein known as leg hemoglobin. Leg hemoglobin or legoglobin. It is a nitrogen or oxygen carrier because naturally occurring oxygen and nitrogen interact similarly with this protein. It is a hemoprotein found in the nitrogen fixing root nodules of leguminous plants. It is produced by legumes in response to the roots being colonized by nitrogen fixing bacteria termed rhizobia as a part of the symbiotic interaction between plant and bacterium. Roots not colonized by rhizobium do not synthesize leg hemoglobin. Leg hemoglobin has close chemical and structural similarities to hemoglobin and like hemoglobin is red in color. The holoprotein, that is protein plus heme cofactor, is widely believed to be a product of both plant and the bacterium, in which the apoprotein is produced by the plant and the heme, that is the iron atom bound in a porphyrin ring, is produced by the bacterium. There is some evidence, however, that suggests that the heme moiety is also produced by the plant. Leg hemoglobin has a high affinity for oxygen, about 10 times higher than that of human hemoglobin. This allows an oxygen concentration that is low enough to allow nitrogenase to function, but not so high as to bind all the oxygen in the bacteria, providing the bacteria with oxygen for respiration. Heterocysts in Cyanobacteria. Heterocysts are specialized pale yellow thick walled cells formed during nitrogen starvation by some filamentous cyanobacteria such as Anabaena spherica. These cyanobacteria have specialized and morphologically distinct cells called heterocysts, which maintain a micro anaerobic environment. Single heterocysts develop about every 9 to 15 vegetative cells. Heterocysts have an additional glycolipid wall which is impermeable to oxygen. Heterocysts are devoid of photosystem II, so the process of oxygen evolution does not take place in them, and hence low oxygen concentration is maintained. Heterocysts have symbiotic relationship with the vegetative cells. They provide vegetative cells with ammonia, which they form by nitrogen fixation, and get carbohydrates in turn from the vegetative cells. Heterocysts have only photosystem I, allowing them to generate ATP by cyclic photophosphorylation. Microorganisms that fix nitrogen. Diazotropes are a diverse group of prokaryotes that includes cyanobacteria, for example, the highly significant Trichodesmium and Cyanothes, green sulfur bacteria, and Acetobacteriaceae, Rhizobia and Frankia. Cyanobacteria inhabit nearly all illuminated environments on earth and play key roles in the carbon and nitrogen cycle of the biosphere. In general, cyanobacteria are able to utilize a variety of inorganic and organic sources of combined nitrogen like nitrate, nitrite, ammonium, urea or some amino acids. Nitrogen fixation by cyanobacteria in coral reefs can fix twice the amount of nitrogen than on land. Around 1.8 kg of nitrogen is fixed per hectare per day. The colonial marine cyanobacterium Trichodesmium is thought to fix nitrogen on such a scale that it accounts for almost half of the nitrogen fixation in marine systems on a global scale. Symbiotic nitrogen fixation. Root nodule symbiosis. The most familiar examples of nitrogen fixing symbiosis are the root nodules of legumes, for example, peas, beans, cloves, etc. Plants that contribute to nitrogen fixation include the legume family Fabaceae with taxa such as clovers, soybeans, alfalfa, peanuts, etc. They contain symbiotic bacteria called Rhizobia within nodules in their root systems, producing nitrogen compounds that help the plant to grow and compete with other plants. In all cases, the bacteria invade the plant and cause the formation of a nodule by inducing localized proliferation of the plant host cells. Yet, the bacteria always remain separated from the host cytoplasm by being enclosed in a membrane, which is a necessary feature in symbiosis. When the plant dies, the fixed nitrogen is released, making it available to other plants and this helps to fertilize the soil. In many traditional and organic farming practices, fields are rotated through various types of crops, which usually includes one consisting mainly or entirely of clover, which is often referred to as green manure. Non-leguminous symbiosis. Although by far the majority of plants able to form nitrogen fixing root nodules are in the legume family Fabaceae, there are a few exceptions. Parasponia, a tropical genus in Cyanobacteria, is able to interact with rhizobia and form nitrogen fixing nodules. Actinorhizal plants such as alder and bayberry can also form nitrogen fixing nodules thanks to the symbiotic association with Frankia bacteria. These plants belong to 25 genera distributed among 8 plant families. Frankia is a genus of the bacterial group termed filamentous bacteria actinomycetes. They form nitrogen fixing root nodules, sometimes called actinorhizae, with several woody plants of different families such as alder, sea buckthorn, which is common in sand dune environments, and casuarina. Here you can see a young alder tree growing in a plant pot, and here you see the part of the root system in this tree bearing the orange yellow colored nodules containing Frankia. Alder and the other woody host of Frankia are typical pioneer species that invade nutrient poor soils. These plants probably benefit from nitrogen fixing association while supplying the bacterial symbiont with photosynthetic products. Cyanobacterial associations. The photosynthetic cyanobacteria often live as free living organisms in pioneer habitats such as desert soils or as symbionts with lichens in other pioneer habitats. They also form symbiotic associations with other organisms such as the water fern Azolla and cycads. The association with Azolla, where Cyanobacteria anabiena azolei are harbored in the leaves, has sometimes been shown to be important for nitrogen inputs in rice paddies, especially if the fern is allowed to grow and then plowed into the soil to release nitrogen before the rice crop is sown. A symbiotic association of Cyanobacteria with cycads is shown here. This is a pot grown plant. Here is a close up of the soil surface in this pot. Short, club shaped branching roots have grown into the aerial environment. These aerial roots contain a nitrogen fixing cyanobacterial symbiont. Rhopelodia gibba, a diatom algae, is probably the only eukaryote with cyanobacterial nitrogen fixing endosymbiont organelles. The spheroid bodies reside in the cytoplasm of the diatoms and are inseparable from their host. Thus, in this module, we have discussed in detail the process of nitrogen fixation. In the next module, we will talk about the regulation of nitrogen fixation and other processes like nitrification and denitrification.